Welcome to the deep dive. Today we're looking at something that is really a fundamental shift in biochemistry, how we design new proteins.
Right.
For decades, if you wanted a new enzyme, a biocatalyst, you're basically stuck with years of just laborious trial and error in a lab.
And then even more time just tweaking it. That time sink is exactly what these new architectures, RIFDiff and RF Diffusion 3, are trying to eliminate.
So our mission for this deep dive is to explore this new frontier of computational protein design.
Yep, and specifically how deep learning is finally letting researchers create these super efficient biocatalysts in what they're calling a one-shot design.
Bypassing, maybe a decade of work.
A decade or more.
We've got two pretty advanced research papers detailing these new methods, and they're not just small tweaks.
Oh, no, these are total architectural overhauls. They're designed to solve a problem, a precision problem that has just plagued the field forever.
So what is that problem exactly?
Well, natural enzymes are they're exquisite machines. They have incredible precision, incredible speed.
And when we try to copy them?
Historically, our initial designs were just slow. So we'd have to rely on this process called directed evolution.
Which is basically just, high throughput screening and mutation, just hoping it evolves into something that works.
Yeah, you're just hoping. Exactly.
But if we look back, the foundational idea behind all this was actually pretty sound. It was based on something called the transition state model.
That's right. The idea is you figure out the reaction's highest energy point, the transition state, and then you design a minimal active site, a theozyme, to sort of hold it and stabilize it there.
Conceptually, that makes perfect sense. So where did it fall apart?
In the execution, it all came down to atomic accuracy. Early computational methods, they could design the concept, but the actual proteins they made were really weak catalysts.
Why? What was the miss?
They just couldn't place the critical amino acid sidechains precisely enough in 3D space. I mean, to be successful, you needed the final protein to match the blueprint with an RMSD that's a root mean square deviation of less than 1 angstrom.
1 angstrom. That's unbelievably small. It's like the diameter of a hydrogen atom.
It is. It's like trying to land a jumbo jet perfectly on one specific grain of sand.
And if you're off by just a tiny fraction?
The whole machine fails, exactly. And the researchers, they learned a really painful lesson from this when they analyzed what happens after those initial designs were put through directed evolution.
Right.
They looked at an enzyme, a retroaldolase, and found that evolution didn't just fine-tune the design. Sometimes it completely replaced the planned catalytic machinery with something totally new.
So the computer's original blueprint wasn't just a little imprecise, it was fundamentally wrong.
Or at least fundamentally incomplete.
Right.
The biggest insight was that evolution gets function not just through putting atoms in the right place, but through dynamic control.
Dynamics. You mean how it moves?
Exactly. Simulations showed that successful evolved enzymes work by making their active sites extremely rigid. They lock those catalytic residues into place, which cuts down on all the molecular noise that would otherwise slow the reaction down a millionfold.
So the challenge wasn't just where do the atoms go?
No.
It became how do we lock them in that exact position and stop them from wiggling around?
Precisely. And that brings us to RIFDiff, the first architecture we're looking at. It was designed specifically to test if we could get that high efficiency in one shot, but by starting with these pre-optimized rigid catalytic centers.
Okay, let's unpack how RIFDiff works. It seems like it had to solve two huge problems.
Yeah.
Placing the machinery and then making sure there was a proper binding pocket.
It did, and with some really ingenious solutions. So for placement, it uses what they call artificial motif libraries.
Okay.
They take the catalytic amino acids they need, and they embed them into these small helical fragments. Then they only select the sidechain orientations, the rotamers, that are energetically stable within that fragment.
So RIFDiff isn't just randomly placing atoms. It's making sure all the critical parts are already pre-vetted to fit together before it even starts building the bigger protein.
That is a perfect way to put it, yes. And then for the binding pocket, previous models would often create these shallow pockets or, have atoms clashing.
Right.
RIFDiff solved this by adding a temporary volume placeholder, an alpha helix, during the generation process.
A temporary void.
Yeah.
So they're designing empty space on purpose.
Yeah.
That feels like a totally new way of thinking about it.
It is, and it's brilliant. They diffuse the protein backbone around this placeholder.
Yeah.
That forces the structure to create a deep void and an entry channel. Then they computationally remove the helix, and you're left with a perfect pocket that mimics natural enzymes.
So let's talk about the results, because the technical side here paid off. Hugely.
It really did.
They used the retro-L delays, or RD benchmark, and the best designs, RAD75 and RAD29. They achieved a 5 million-fold rate acceleration.
A 5 million-fold acceleration. I mean, that is what you strive for in design. And to give you some perspective, that activity surpassed a really famous benchmark, the catalytic antibody 38C2, which took a ton of lab evolution.
So RIFDiff did in one go what used to take years of optimization.
Exactly. And the products were clean, too. The designs were incredibly stable. Most stayed folded above 90 degrees Celsius, and they were highly selective. RAD35 got 99% in antiomeric excess.
You get almost perfect purity.
Almost perfect. And they showed it wasn't just for redesigning old reactions. They designed for the Morita-Baylis-Hillman reaction, which is a carbon-carbon bond formation that nature isn't very good at.
And what happened?
One of their designs was 1.5 times more active than a variant that had already gone through 8 rounds of directed evolution.
So RIFDiff proved the main idea.
Yeah.
You can scaffold these super active enzyme arrays in one shot.
Yep.
But, and here's the twist. This is where the scientific mystery comes in.
This is the critical limitation. The structural analysis was so revealing, they found that some of their slower designs, like RD13 and RAD17, had near atomic precision. I mean, their backbone RMSD was under 1.2 angstroms.
So they were super close to that 1 angstrom target.
Very close. And yet, these designs were 100,000-fold slower than the benchmark.
And that's the paradox, isn't it? How can such a tiny difference in the static picture lead to such a catastrophic failure in function? Was the active site just vibrating too much?
That's exactly the right question. It shows that just measuring the structural precision of the enzyme by itself, the apostate, is not enough. The real problem was flexibility. Molecular dynamic simulations confirmed it. The designed active sites were just way more flexible than the evolved ones. That key catalytic lysine in RAD36, it was constantly just drifting away from where it needed to be.
The protein wasn't holding its shape long enough to do the work.
Correct. The rigidity was gone. And here's where the story gets even better. They looked at the crystal structure of one of their most successful designs, RAD32.
Okay.
And they found a planned catalytic residue was actually in the wrong position. It was unproductive. But the protein compensated.
The protein fixed the computer's mistake on its own.
A nearby tyrosine residue, one that wasn't even supposed to be part of the core catalytic machine in the blueprint. It just sort of stepped in and did the job of the mispositioned one. The protein's chemistry was smarter than the design model.
That completely changes how you have to think about success. So if the static blueprint, the RMSD, is basically useless for predicting activity, what metric did work?
The metric that worked came from modeling the dynamic interaction. They used tools like AlphaFold 3 to model the enzyme-substrate complex. They weren't looking at the static protein anymore. They were looking at hydrogen bond distances and how the substrate was positioned during the reaction's intermediate state.
So success isn't static, it's dynamic.
It is entirely dynamic.
So if RIFDiff taught us we need atomic precision and dynamic control, how does the next generation RF Diffusion 3 actually give us that control?
RFD3 is the technological leap that was needed. See, previous models worked at the residue level. They modeled the backbone, but the side chains were treated more generally. RFD3 switches to a true all-atom process. It explicitly models all 14 or so atoms per residue.
That sounds computationally astronomical. But what does that actually let a designer do?
It makes specifying constraints so much easier. Since every atom is modeled, you can set precise geometric rules. This hydrogen bond donor must be this far away. This atom has to be buried this deep. You get incredible fidelity.
And the big new feature here is co-diffusion. This seems like a major change for designing complex systems, right?
It changes the game entirely. Co-diffusion means RFD3 can generate the protein structure and the conformation of other things, like small molecules or DNA, all at the same time.
Which is critical if your target isn't rigid.
Exactly. Previous models really struggled with that.
Let's talk about the validation then, because RFD3 seems to go way beyond just enzyme scaffolding.
Absolutely. They used co-diffusion to design a sequence-specific DNA binding protein, DBRFD3. When they tested it, it showed low micromolar binding affinity.
And for you, the listener, that low micromolar affinity is really important. It confirms that RFD3 is building things that actually work like natural systems in a real biological context.
And for classic enzyme design, RFD3 proved its control there too. They scaffolded a cysteine hydrolase motif and got an enzymatic activity that just blew away previous designs for that same reaction. It just shows superior control.
Okay, so let's step back for a second. RIF-DIF proved we can build highly active enzymes in one shot.
Right.
But it also gave us this damning evidence that the static structure is a terrible predictor of success. The real challenge is managing dynamic flexibility.
And RFD3 is the field's technological answer to that challenge. We've moved from just, hoping the protein folds correctly to being able to precisely command how the active site is built, how it's buried, and how it interacts with the substrate atom by atom.
The power is really shifted from the lab bench to the computing cluster. We have the tools, but we're still limited by our ability to predict that complex conformational dance of the protein.
Exactly. The ultimate hurdle, the one that's still there, is that predicting high enzymatic activity before you do the experiment, that remains incredibly challenging. The design itself is no longer the bottleneck. We can now build almost anything we can imagine.
So what does this all mean for the future? If RIF-DIF and RFD3 have made the engineering part routine, if we've solved the how to build it problem, where do we go from here?
The major roadblock just shifts entirely. It moves from building the catalyst to knowing how it works. For a truly novel reaction, something nature has never seen, the biggest challenge might soon be defining what the ideal catalytic array even is.
So we need a better understanding of the chemistry itself.
We need a mechanistic understanding of chemistry that goes far, far beyond what we currently have. We can build anything. We just have to know exactly what sequence of atoms, hydrogen bonds, and dynamic motions will make it work. The limit is no longer engineering. The limit is knowledge.